Hello, everybody. Thank you so much for joining us for today's Neuroimaging Summer Series, showcasing the cutting-edge research of the neuroimaging faculty and associates at the Michigan Alzheimer's Disease Research Center. My name is Stephanie, and I will be moderating things here on the back end. Later on in the talk, I will be sending out a Qualtrics evaluation, so please take the time to fill out the evaluation. We really value your opinions and want to know what kind of talks you are looking forward to hearing from us next. Mark your calendars for the next neuroimaging series. It will be on July 15th at noon. Dr. Patrick Pruitt will present brain connectivity in cognitive aging, a primer on concept, measurement, and application. And with that, I am honored to introduce our group that's speaking today. These presenters are part of a collaborative and interdisciplinary group focusing on quantitative MRI neuroimaging at Michigan Medicine and Michigan Engineering. Dr. Navid Seraji is a clinical associate professor in the Department of Neurology and studies non-invasive methods for monitoring disease progression in neurodegenerative disorders with the ultimate goal of developing tools to monitor the efficiency of therapies in neurodegenerative disorders. Dr. Jon-Fredrik Nielsen is a research associate professor in the Department of Radiology and Biomedical Engineering and is currently working on improved methods for functional MRI, vendor agnostic pulse sequencing program, and quantitative MRI. Amaya Murguia is a PhD student researcher in the Department of Electrical Engineering and Functional MRI Lab, and is interested in developing faster MRI image acquisitions and reconstruction techniques for quantitative MRI data and optimizing these methods. And without further ado, I'd like to turn things over to Dr. Navid Seraji, who will be starting us off today.
Okay, thank you, Stephanie, for the introduction, and to also Brooke and the rest of the MADRC folks for arranging these neuroimaging summer series, as well as to everybody who is in the webinar today as participants, and a warm welcome to also those who will be hopefully viewing this series in the future. Throughout the presentation here, we'll be presenting some of the outline of what we're talking about in different sections. I just wanted to start out by sort of talking about the title as an introduction. We're going to be talking about mapping myelin content with MRI and looking at these existing and emerging methods. So before we do that, it's sort of important to say, why do we even care about myelin in Alzheimer's disease and related disorders? So the sort of the departure of this webinar, the point of departure for this webinar is this assertion that Alzheimer's disease and related disorders have a reciprocal relationship with white matter disease. In other words, in patients with Alzheimer's disease, white matter tracks, the myelinated axons, are not healthy, and they show several pathological changes. And in turn, these changes, these pathological alterations in white matter disease also has an effect on disease progression, on neurodegeneration, as well as acceleration of tissue loss. The reason I bring this up is because Alzheimer's disease has been always thought as a cortical problem, a cortical dementia. And that refers to the fact that the structures in the brain that are mostly affected are in the cortex, most prominently in the parietal lobe, as well as in the hippocampus. And as they undergo atrophy and volume loss, they affect the function of the cognitive functions that are associated with these areas, including loss of episodic memory, semantic fluency, and visuospatial function. And this concept is juxtaposed to the subcortical dementias, which traditionally involve areas of the brain in the subcortical region, namely the white matter tracts, the thalamus, the basal ganglia, and so on. And they often have a different cognitive profile, which includes a decline in processing speed, sustained attention, executive function, and so on. However, we are finding out more and more that there is a commonality between these two sort of different causes of cognitive decline. Both Alzheimer's disease and cortical dementias, as well as the subcortical dementias, or what used to be called subcortical dementias, have an alteration in cerebral blood flow. There is a reduction in cerebral blood flow. There's alterations in the blood-brain barrier, and the whole neurovascular unit, including astrocyte and feed processes and microglial inflammation and other pathological processes are common to both types of disease. So going a little bit more details, Alzheimer's disease affects white matter. Alzheimer's disease affects myelin. The evidence for this claim comes from in vivo and postmortem studies that have shown myelin and oligodendrocyte degeneration in brains of patients with Alzheimer's disease. Also in tissue, in cortical tissue, in postmortem studies, genetic studies have shown a prominent dysregulation in the oligodendrocytes gene expression. And as we will see later also, in vivo studies have also shown myelin damage that's associated with presence of beta amyloid and tau pathology. Now, looking at the other side of the arrow, white matter disease also affects progression of neurodegeneration and cognitive decline in patients with Alzheimer's disease. This sort of a sequence of events of myelin degeneration results in impaired microglial function and impaired beta amyloid clearance. And in addition, the sort of the temporal progression of changes suggests that there is early involvement of myelin pathology, even preceding accumulation of tau pathology. And sort of an indirect circumstantial evidence of the myelin pathology worsening Alzheimer's disease is the fact that ApoE, in particular ApoE4 gene, which is involved in the lipid transport and homeostasis and myelination, is a well-known risk factor for earlier development of Alzheimer's disease. So I'm hoping that by now we have enough evidence to sort of believe that myelin and white matter is an important thing to study in Alzheimer's disorders. One of the issues, however, is that, okay, if we want to study something, we have to have a good way of interrogating it, measuring it, and monitoring it. Probably one of the problems here is that even though we've had a significant advances in imaging and looking at Alzheimer's disease, are the main techniques that we use in the large clinical studies or even in the clinic itself dates back to about 40 years ago as to what we had at our disposal for looking at white matter tracts and demyelination. In 1980s, it was noted that patients with Alzheimer's disease, their MRIs looked different than healthy persons. In particular, the T2 weighted sequences where white matter tracts are usually are more hypointense and are darker, had an alteration in the signal where they appear to have a higher intensity and appear to be white. So these white matter hyperintensities were associated with some form of pathology in the myelin. And the grading system, Fazekas grading score, was started around 1987, which looked at the extent of these white matter alterations. The score is basically a subjective, semi-quantitative scoring of what is the extent of white matter alterations around the ventricles, as well as in the deep white matter. The more these white matter lesions coalesce together, the higher the score is. But I'd like to present two cases as an illustration of the difficulty with Fazekas score, and in general, looking at the extent of white matter hyperintensities as a measure of pathology. This is a case of a 75-year-old woman who presented to the clinic with concerns about memory loss, and the MRI is shown on the right-hand panel here, where the T2 flare sequences show these hyperintense lesions in the white matter, and they're starting to coalesce together from these punctate regions, sort of gathering together to form these larger areas of white matter involvement. So the Fazekas score for this patient would be a total of 4. And correspondingly, this patient demonstrated difficulty with retrieval of memory, with executive function, and so on, which was significantly below average and is consistent with these changes. However, if we look at the next patient, an 83-year-old woman who also was concerned about memory loss, and her MRI showed a far more extensive involvement of the white matter tracts. And as you can see, there is encroachment of the periventricular white matter changes into the subcortical areas. It's hard to find punctate regions anymore. They've all coalesced into large patches of white matter involvement, and her Fazekas score was 6. However, her cognitive profile actually was normal or above average in all the measures that were affected in the previous patient. So this is the main difficulty with using the extent of T2 changes in trying to correlate with the disease state and symptoms. So if the extent of T2 hyperintensities doesn't correlate well with cognitive deficits, one could sort of conclude, or at least make the assertion, that perhaps some of the areas that appear abnormal in these T2 images are actually not representative of a sort of a malignant pathological process. In other words, some of these white matter areas may represent just a more benign process, such as tissue edema and so on, whereas other ones will be more reflective of tissue destruction and cellular loss. In other words, not all the white matter lesions are the same. So we would like to, explore this assertion a little bit more and try to find or talk about techniques that try to make the alterations in the MRI more specific and find a signal that can distinguish some of these benign processes from perhaps more significant ones. So in the next section, I think Amaya will talk about some of the MR techniques that are currently available and what makes them potentially more specific and how they have been used in patients with ADRD. And later we will review some of the more aspirational techniques that are very promising, although they're not yet available for in vivo studies. So with that, I'll give up my control and pass it to Amaya.
Thanks, Navid. All right. So as Navid said, clinical T2 MRI scans are not specific to a particular pathology. So for example, this T2 hyperintensity could have been caused by inflammation, myelin loss, axonal loss, or other causes. To try to better understand which process is happening, we turn to quantitative MRI. So we will review 3 MR methods that will give us a more quantitative myelin metric: diffusion MRI, magnetization transfer, and myelin water imaging. Diffusion MRI is one of the main techniques that we have for investigating white matter. It measures water molecule diffusion and its directionality. For example, there is faster diffusion along the length of an axon, but slower diffusion perpendicular to the axon. Axons, myelin, and glia can all hinder diffusion. Diffusion tensor imaging, or DTI, is a quantitative method to measure the amount of diffusion in a certain direction. It models the diffusion process as a Gaussian process. Fractional anisotropy, or FA, is a metric that comes out of DTI modeling that is higher for more restricted diffusion, and mean diffusivity, or MD, is one that is lower for more restricted diffusion. A study came out in 2022 that used diffusion tensor imaging to look at white matter damage in Alzheimer's disease patients. In this study, both MD and FA values for a patient were plotted versus the patient's mini mental state examination score, where a higher score corresponds to better cognitive ability. The gray rectangles represent mean values plus or minus 2 standard deviations for cognitively normal controls. The MMSE scores correlated negatively with MD values and positively with FA values. Both metrics in this study indicated that better white matter integrity in the brain correlates to better cognitive ability. Although diffusion MRI can give us information about white matter tract integrity, which is influenced by myelin, it does not give us a quantitative measure of myelin content or the amount of myelin present. On its own, diffusion MRI cannot provide us with enough information about myelin in the brain. So next we turn to another popular technique for imaging white matter called magnetization transfer, or MT. MT involves the interaction of protons in the macromolecules, such as myelin, and protons in the free water. The protons on the macromolecules have very short T2 values, less than 1 millisecond, so we cannot directly collect the signal fast enough with MRI. The free water molecules have a much longer T2, and so they're MR visible. We can apply RF energy via an off-resonance MT PrEP pulse exclusively to the macromolecular water protons. And then some of this energy is transferred to the free water pool via dipole-dipole interactions. Then we can image the free water pool and gain information about its interaction with the macromolecular pool. Also, the MT effect also influences the observed T1 value. The magnitude of the MT effect can be quantified by obtaining 2 images, one with the MT PrEP pulse and one without it, and then subtracting them. The magnetization transfer ratio, or MTR, reflects the difference between these 2 measurements. For example, the MR signal from white matter is more suppressed than the MR signal from gray matter, causing white matter to have a higher MTR than gray matter. Other MT metrics include the pool size ratio F, which is the ratio of the number of spins in the macromolecular pool to the number of spins in the free water pool, and also inhomogeneous magnetization transfer, which is similar to MTR, but obtained from MT experiments that have been designed to be more specific to myelin. This next study looked at MT imaging of Alzheimer's disease patients. And this study used MTR to measure the amount of myelin present, where a higher MTR corresponded to more myelin. The researchers found that the MTR signal in the temporal lobe decreased with Alzheimer's disease duration, indicating that the amount of myelin present decreased as the disease progressed. They also found that MTR increased with higher MMSE scores, indicating that the amount of myelin present is positively correlated with cognitive ability. Additionally, a discriminant analysis was nicely able to separate the AD and control groups using the MTR signal. Although MTR is a useful metric for studying myelin content, it is not a quantitative myelin measure and is based on the relative contrasts of the images with and without the MT pulse. In addition, MT scans are long and provide information about interactions between water molecules and macromolecules, and so is not specific to the myelin macromolecule. Inhomogeneous MT imaging is a little more specific to myelin, but these scans are even harder to do in vivo due to the long scan time. So next, we'll discuss myelin water imaging, which is a quantitative MRI measure specific to myelin. On the left is an electron micrograph image of myelin. The water molecules trapped in the myelin sheath, shown in blue here, are more tightly confined than the water molecules in the intracellular and the extracellular space, also called the free water, which is shown in purple. Due to being more tightly confined, the water molecules have a shorter T2 value than the free water molecules. In the graph on the left, the decay curve with the blue dots shows the MRT2 signal decay for a substance with one water pool and thus one T1 component. This signal decay is mono-exponential. The decay curve with brain images on top here shows the MRT2 signal decay for a substance with multiple water pools and thus multiple T2 components. And this signal is multi-exponential. In classic myelin water imaging experiments, a multi-echo spin-echo scan is conducted, and a curve is fit to the acquired multi-exponential data using non-negative least squares regression to determine the relative amount of signal coming from a particular T2 value. The plot on the right shows the relative amplitudes of the signal from the various T2 values. There are two peaks, one at lower T2 values, corresponding to the myelin water, and one at higher T2 values, corresponding to the free water. The area under the myelin water curve divided by the total area under the curve is defined as the myelin water fraction, and this is the measure of myelin content. This grayscale image shows a brain myelin water fraction map, where higher values correspond to brain white matter with more myelin and a higher myelin water fraction. Although the multi-echo spin-echo scan is the traditional scan used for myelin water fraction mapping, the data acquisition process is very slow. Many groups, including ours, are working on faster methods for myelin water fraction mapping to make them more feasible for clinical use. These results show a myelin water fraction map generated using a fast, steady-state MRI pulse sequence and processing method developed in-house at Michigan. This map is from a 71-year-old cognitively normal female volunteer in the Memory and Aging Project study. The myelin water fraction map shows reduced myelin water fraction and significant myelin loss within the T2 lesions that can be seen in the T2 flare scan. A 2022 study looked at both myelin water fraction and tau pathology in patients with Alzheimer's disease. In this graph from the paper, myelin water fraction is plotted on the x-axis and tau PET scores are plotted on the y-axis. The two left graphs show controls, and the two right graphs show Alzheimer's disease patients with two different data sets. All data was taken from regions in the cortical gray matter. The tau PET score and myelin water fraction values were shown to be negatively correlated, meaning that higher myelin water fraction and more myelin corresponded to less tau. They also found that reduced myelination along fiber tracks corresponded to increased tau spreading along these tracks. Although myelin water fraction is more specific to myelin content than the other methods previously mentioned, some drawbacks to its use include the long scan times, non-trivial processing for the generation of these myelin water fraction values, and the low signal-to-noise ratio of the myelin water fraction maps. Another challenge is that we don't have a good ground truth for these maps, so it is difficult to determine their accuracy. Histology has been studied as a method to validate how well the maps correspond to myelin content, but histology studies cannot be conducted in vivo. So we have reviewed a few popular quantitative MRI methods for looking at myelin content in the brain: diffusion MRI, magnetization transfer, and myelin water imaging. But we should note that there's other methods as well. If you're interested, you can check out this review paper, which is a great way to learn about more of these methods. Each is useful in its own right and probes a different aspect of myelination in the brain. They have the potential, when used together, to provide complementary information about myelin content in brain tissue and differentiate between different types of brain lesions. However, as previously mentioned, some important issues are slow acquisitions, lack of specificity to myelin, and limited ground truth validation. As I mentioned before, the best method we have is correlation with histology, which is the motivation behind our current study, which I will be talking about next. So our current study involves conducting both high-resolution MRI and histological processing of post-mortem brain tissue samples from the MADRC brain bank. Since one MR method is not enough to fully characterize a tissue sample, we conduct a suite of MRI imaging experiments on these tissues, and also a variety of histological tests to identify the pathology associated with a specific lesion in the sample. We aim to discover if there is a mapping between the imaging space and the histology space. Our workflow in this ongoing study involves tissue selection using pre-mortem clinical T2 scans, tissue preparation and MR acquisition, followed by tissue fixation, histology, and immunohistochemistry, and quantification of histological measures. We employ the standard methods with long acquisition times since we are conducting our study on ex vivo samples. However, before investigating this mapping, we needed to make sure that the tissue preparation techniques minimally altered the tissue and affected the results. The MADRC contains tissue samples preserved in two different ways: frozen and formalin-fixed samples. We conducted a preliminary study to decide whether to proceed with scanning the frozen or fixed MADRC samples by developing our protocol with sheep brain tissue samples. We scanned the same sheep brain sample when it was fresh, frozen and subsequently thawed, and formalin fixed using various quantitative MRI methods, and then conducted Luxel fast blue staining on the sample where the myelin fibers appear darker blue. The images here show fast spin echo images of the same coronal section of an ex vivo sheep brain when it was fresh, thawed, and fixed and stained with LFP. Next, we investigated whether the freezing and thawing and fixation of the tissue affected the MR signal compared to the fresh sample. Here is our myelin water fraction and MT results for one of the sheep brain tissue samples. The first column shows the MRI image of the same sample that was scanned fresh, thawed, and fixed. The red box is a gray matter ROI, and the blue box is a white matter ROI. The second column shows the multi-exponential signal decay and fit to the data for one voxel in each of these ROIs. The third column shows the relative amplitudes of the various T2 components in the white and gray matter ROIs. The fourth column shows the myelin water fraction maps for each sample preparation. We observed that the myelin water fraction values were higher in the fixed tissue sample compared to the fresh and thawed. We also conducted magnetization transfer experiments on the tissue samples and observed that MTR values were higher in the fixed tissue as well. Potential explanation for this finding is that the formaldehyde added for the fixation immobilized more macromolecules or created more pathways for magnetization transfer. We also looked at the correlation between myelin water fraction and MTR values with Luxel Fast Blue histological results. We plotted the average myelin water fraction and MTR of both white and gray matter ROIs versus LFB. The green points are from the fresh sample, the orange points are from the thawed sample, and the purple points are from the fixed sample, all from the same ROIs in the sample. We found a positive correlation between the MR and histology results, found consistently higher myelin water fraction and MTR values in the fixed tissue compared to the fresh and thawed. Tissue fixation seemed to affect the myelin water fraction and MTR results, so we plan to scan the thawed MADRC human brain samples moving forward. And now I'll turn it over to John to describe some emerging imaging methods.
All right. Thanks so much, Amaya. So I will be talking about a couple of what we might call emerging methods that's afforded to us by the ability to scan long time at high field. So as Amaya described, we have a workflow now for obtaining high resolution MRI data in tissue samples. We want to take advantage of that to explore alternative and potentially more advanced MRI methods that take longer to run, but that might give us more information about the tissue state. And I'll describe the two such methods. And the first one is the bioexponential T1. So let me motivate that. So most of us, we quote unquote grew up learning MRI and learning that longitudinal magnetization recovers at an exponential rate. Described by a single time constant T1. Well, that's actually slightly wrong. If you perform an inversion recovery experiment, as shown here, you'll find that the recovery is actually bi-exponential. So these images show an image, an EPI image obtained after inverting the longitudinal magnetization, and then waiting some time Ti, and then acquiring the EPI image. And so each circle here represents an image, and we plotted here the signal in a white matter ROI and a gray matter ROI. So what we can observe is that the recovery is actually bi-exponential. So if you look closely here, so the fit here on the top is done with a bi-exponential model, and so then we get a very good fit. If you do mono-exponential fit, you'll see that there's some deviation here at very slow time. So it has a fast time constant at less than about 100 milliseconds, as well as a long time constant. And so the classic kind of understanding of what's going on here is that the observed T1 is driven by dynamic MT effects. And the idea is that the inversion pulse, it inverts water, but not macromolecules because of the very short T2. And that causes a rush of magnetization into the water during the first 100 milliseconds or so until things equilibrate. And then relaxation or recovery from that point on is mono-exponential. And so what we're doing is that we're obtaining data like this and fitting it to the following model shown here. So the signal is a sum of two exponentials. So S refers to the slow component, slow relaxing component. F refers to the fast relaxing component. So our overall goal here is to establish a workflow for quantitative T1 analysis based on this approach, and then correlate that with the histology. And this has a couple of potential benefits. One is that these model parameters are quantitative tissue parameters that should be more specific to the state of the tissue than just conventional T1 weighted images. And it's also based on a widely available sequence, so it's usually available on every scan. Okay, so once we fit the data to the model, what do the model parameter maps look like? So let's first look at the amplitude of the slope component, shown here in green. So that's obtained mainly from the later time points in the data, where we see that the fit is really good. And as I said, in this regime, it really does make sense to talk about mono-exponential T1. And we can clearly observe gray and white matter contrast, and the corresponding slow T1 maps also have this property. And so recall that these are quantitative tissue parameter maps. These maps should not depend on the details of the MRI sequence that was used to acquire it, unlike a lot of other conventional T1 mapping techniques. And so we think that perhaps the amplitude and the slow T1 are good candidates for correlating with histology and for comparing lesions versus healthy tissue. We can also look at the short component maps. They're shown here in the second column. The amplitudes here are a lot smaller, and also the fast T1s are quite short. And so on the whole, this means that these components, they explain a relatively small part of the observed signal, but it doesn't matter at those short inversion times as I showed. Perhaps these are too noisy to be immediately useful, but that remains to be determined. And so we can also calculate the fraction of the fast component relative to the total combined amplitude. And this is perhaps a semi-quantitative metric for myelin volume or weight fraction. And we can obtain these with reasonable image quality. So these are some of the parameters that we can get out of this that are specific to the tissue. So in addition, we're also exploring using this data in another way for segmenting and classifying the tissue. And we think this may be useful for our analysis pipeline. And so the idea here is to first create a 2D histogram, that's shown here on the top right, with total amplitude on one axis. So this is the amplitude of those A coefficients. And on one axis, and then the slow or long T1 on the other, as shown here. And so we see that the data tends to cluster into two groups, and that we assign to gray matter, circled there in red, and then white matter in blue. And we assign the voxels in between to be quote unquote mixed. And we see that this gives plausible tissue segmentation maps that give us some confidence that a voxel or ROI is in either pure white matter or pure gray matter, or perhaps in some combination of them. And we've observed that by segmenting or classifying in this 2D parameter space, we get better separation than if we tried to do it just along one dimension. So for example, if we try to base it just on the T1 dimension, we see that there's more overlap, and perhaps it might be more difficult to identify or classify these tissues. Okay, so that's the T1 analysis. The second topic is multidimensional quantitative MRI, and we're focusing on diffusion T2 relaxometry. So this is based on a very different way of thinking about the data than what you may be accustomed to. So let me start with what you already know. So let's talk about T2 weighted images. So one T2 weighted image would be represented here by one red circle. So this means that you dialed in your echo time in your scanner and you acquired an image that represents one acquisition. So I'm going to refer to one such acquisition or one circle as a zero-dimensional MRI experiment, because we've only acquired 1 point sort of along this TE dimension. So the next level of complexity might be to acquire 2 or more T2 weighted images with different TE. So acquiring these several images now by varying the TE and acquiring multiple images. So now we can, for example, fit that data to a model, and we can calculate the T2 in this one, and I'll refer to this as a one-dimensional experiment, and that's commonly done. So if you have many more than 2, we can try to calculate a T2 spectrum, just like Amaya showed, which is what we do in Myelin water fraction imaging. And we can do this for other MRI contrasts as well. For example, to obtain a T1 measure, we can then, just like I showed, we can vary the TI time. So then we can calculate the parameters that I just talked about. Or we can do it potentially along the diffusion dimension as well to get a diffusion spectrum. So, let's see. All right, I'm gonna make sure I have the right notes here. Okay, so the idea behind diffusion relaxation MRI, or more generally, a multi-dimensional MRI where you go beyond one dimension, is to acquire data along 2 or more dimensions. So for example, we could acquire multiple images in this diffusion T1 space or TI space. We can acquire, for example, a grid of images, each with a different combination of a B value that you dial in on the scanner and the inversion time. And so this, of course, takes a lot longer, but that's certainly an experiment that you could do. Or we can do the same thing along the diffusion and the echo time dimension, which is what we're focusing so far in this project. But that's just one possibility. So however, when you do this, of course, this takes a lot longer to acquire. So the question is why, is it worth it, or might it be worth it? So the key motivation for doing this is illustrated here on the right. So each figure here shows, you can think of it as a voxel containing either 2 or 3 compartments. So the voxel on the left contains 2 compartments, one with a volume fraction of 25% here with a low T2 value and a high diffusivity, and then the other larger compartment over here. And now suppose you did a 1D measurement along either the diffusion dimension or the T2 dimension to characterize this tissue. And you would see both peaks, and that measurement would accurately represent the 2 compartments. Now suppose you have a voxel now with 3 components, as shown here, this 25%, then this and this. If you did those same 1D experiments, you would in fact obtain the essentially the same data, except for noise, of course. So from these 1D measurement, you would not be able to resolve those 3 compartments. So that's the primary motivation for doing these kinds of 2D measurements. And so the potential strengths can be summarized as follows. You can resolve potentially multiple tissue compartments and potentially look at the coupling between them, and perhaps tailor the acquisition to bring out some components that you care about. Of course, in the long run, we'll need to undersample the acquisition to make this actually practical. But for now, we're quote unquote fully sampling and characterizing this method and getting some experience with it. So here's an overview of how we do this. So we first develop a signal model that describes the data, and we do that using block simulations of many different tissues with a range of diffusivities and T2 values, like maybe 30 by 30. And then we model the acquired data as a sum or superposition of all of those tissue types, with each tissue type or component being scaled or weighted differently by some scalar value. And so our task here is to determine the values of these scalar coefficients. And we do that by fitting this model to the data. And what that's going to produce is what you might think of as a 2D histogram that's shown here. So we're working in the T2 diffusivity space now in these 2 dimensions. And the intensity here basically represents the amplitude of each of those components. So that the goal is to take that 2-dimensional data, fit it to our model, and then obtain 2D histograms like this. And so here's some first results. We're really just getting started with this. So the image on the top left shows a T2 weighted image and the location of an ROI in white matter. And the average signal in this ROI is plotted here on the right. So what I'm showing here is all the data from the 2D experiment collapsed into one vector that we're just plotting. So this first fast decaying curve here is for a single B value and then multiple echo times. And then the next little decay curve here is for a different B value and so forth. So this is just to illustrate that we're able to fit the model and get a good fit to the data. So here, the circles are the data and the blue line is the fit. And the fit for this particular data set is shown in the 2D histogram shown here. And we think there are a couple of things that are interesting and hopefully real. So one is the free what we think is a free water diffusion peak at T2 a bit less than 100. And we observe this, in pretty much all the ROIs that we've looked at. And there's also interestingly what appears to be a restricted diffusion compartment that seems to have a slightly longer T2. We're not sure if that's what that corresponds to, but that's what the data suggests. So that's already intriguing. So also shown here are fits to 1D subsets of this data. So for example, if you do a 1D fit of the data for a particular B value, so just analyze basically one of these decay curves, you can obtain a T2 histogram, and we see a main water peak that's in good agreement with the data that Amaya showed. The smaller peak here is probably an artifact of the numerical fit because of the way the data was acquired, but those are the things that we are looking into. So another thing to note here is that when we perform these fits, the regularization constant or the Tikhonov regularization constant that we use is slightly different. So that also makes the direct comparison between these 1D histograms and 2D histograms a little challenging. So for the diffusion, it's a little busy here, but basically you can look at that red curve to see that we are picking up, those same components. But again, from these 1D profiles, it would be perhaps difficult to discern what the underlying compartments are. And the lower left figure just illustrates how we picked the Tikhonov regularization parameter based on the cross-validation approach. So to summarize what we've done here, brain myelin content is important, we hope, to Epkinus-2 in the study of DRD. Several different MRI contrasts have been proposed over the years, and this is still very much an active area of research, the diffusion, MT, myelin water fraction. In fact, even just within myelin water fraction, that's a field of research on its own, and it's still not clear, which one is the winner there. MESI is still arguably the gold standard, but perhaps not the ground truth. A lot of these methods are complementary. They probe different aspects of myelin and white matter, and so that is a potential source of power that we can utilize. A lot of them suffer from long scan times, and as Amaya mentioned, it can be difficult to validate them. And so we hope to convince you that this direction of research correlating various quantitative MR metrics with histology results may offer a potential solution for validating these approaches. And more advanced MR methods might be able to give us additional information about the myelin content of these tissue samples and other properties of the tissue. So with that, I'll thank you very much for your time, and this acknowledgement slide and the rest of our team. Very much.
Great. Thank you very much. We can open things up for questions. We've got about 10 minutes now. You can put your questions either in the Q&A or the chat, or if you want to say something, raise your hand and I can give you audio access as well. All right. All right, I have one right here. It says, great talk. Can you speak about the potential utility of other MWI metrics, such as Geom T2 mean?
What was I missed can we pull up the chat so I didn't quite catch the last word there.
It says,
geometric to me.
Yep.
I see. maybe that's a better question for Navid. So right now we're focusing on validating the basic method and eventually correlating with histology. And we've done some work on developing faster methods, focusing on the fraction, and we've designed those acquisitions to uh, optimize the estimate of that fraction. But that's actually a very, very interesting point. We have not really actually considered that. So I would say it's something that we probably should add maybe to our suite of metrics as we look at this data. Yeah, that would be my comment. So thanks a lot for this suggestion.
Yeah, and I would just add that, I mean, as you sort of mentioned, there are many metrics out there, and it's going to be impossible to sort of design a study that includes everything. And that this was part of wanting to present sort of this talk, that this really is a collaborative effort that we try to acquire the data and then later on provide it. We want it to be available for others to do different kinds of analysis. We won't be able to do all the MR experiments that generate the entire set of data. But we want to open this up to folks who may say, oh, well, why don't we do this? Why don't we do spectroscopy? Why don't we do this? Why don't we do this other technique? Or if you're using diffusion, we didn't talk about the diffusion experiments we're doing, but why don't you add this higher B value or various parameters? And we're open to sort of receiving that feedback, so that as this project goes forward, as these histological validation projects go forward, they're included in there so that researchers can use the data however they want and model it however they want.
Any other questions for the team? Well, if not, I think we'll end just a couple minutes early. I did put the evaluation in the chat, so you can see that there. You can go ahead and answer that, or it will be coming out in a follow-up email tomorrow as well. So thank you so much for joining us today. It was a really great talk, a lot of really excellent information.
Thank you
So much.